So even though we've talked about how it's ubiquitous outdoors, you cannot take a breath without taking in 1.3 billion of these puppies, or 1.1 trillion of them in an hour. But still, we would still want to show that this is safe for use indoors. So the Food and Drug Administration reviewed air purification devices and said that hydroxyl devices were considered safe based on the prevalence outdoors in nature. So those statements are in that we have those. The Environmental Protection Agency, the Center for Disease Control, and National Institute of Health consulted their databases, which are massive, and there was no data anywhere indicating that hydroxyl use was unsafe. It's very tough to get a government agency, and as a matter of fact, it won't happen. They will not give a statement that says this is safe. They'll say at this time, this is what we're saying, and that's as good as it gets, or a letter of no objection. They also mentioned those three entities, the EPA, CDC, and NIH, indicated that no clinical studies were required. We did them anyway. So we did live animal studies. We also asked the National Institute of Environmental Health Sciences, and that PubMed, a National Library of Medicine, is the largest in the land. And the quote from Colleen Chandler, which is the Communications and Public Liaison for that office, said there was no hard science or research indicating that hydroxyl radical generation is harmful to humans. I like this next part. That applies to both atmospheric and man-made generation. So we don't have to worry when we go outside and we inhale and we take in that 1.3 billion in a breath, and the same thing applies to us indoors. So we went ahead, as I said, and did the live animal testing. And also along with that, first we had to prove what we had, right? So just to have a sticker on your machine that says that your hydroxyl generator, is that an accurate statement? So let's back up the truck a little bit. So we did the world's first. Even NASA hasn't done that, which is kind of interesting, so I have to blow the horn a bit there. The world's first hydroxyl generator toxicity study. That was a rigorous, good laboratory practices, so GLP standard. 13-week study, 60 rats in total were part of the study. 40 were the test group, 20 were the controls. And they were exposed to higher than normal levels of hydroxyls, because we can monitor that. And the nice, interesting thing was that it wasn't just an 8-hour study and they got a rest. They were exposed to this 24/7. So they were checked on the behavioral, toxicological was checked, the gross cellular pathology. Anything that would contact it immediately, which would be the eyes, the sinuses, the mucous membrane, the lungs, the nervous system. All of this was verified. And through all of that testing, there was no morbidity or mortality noted. The tested animals showed no adverse effects. There was nothing wrong with their histopathology, which is their cellular differences. The interesting thing about someone that's watched rat behavior for years and years and years, in fact decades with this lab, was that they noticed that the treated animals were more social, they were more active and more responsive. So when you take a group of animals that are in their own waste, and that creates a certain behavior pattern, they're not their usual selves. But here, these other ones, we got rid of the fecal odor and the urine smell, and they act as if they were an outdoor animal. In other words, they were inquisitive, they were looking at the researchers, they wanted to know what was going on, whereas the control animals were just left in that situation where things were not cleaned up. They were not social, and so they acted like they had seen lab rats for decades. So that was an interesting note, that when you clean up the environment, there's a difference, a behavioral difference. And so when we want to see that, wherever in our workplace, if we're working in a sewage treatment plant or something where it's very pungent, to clean up those gases would certainly have a net effect on us as well. Very interesting on that tox study. When we create what we do, the hydroxyl, we have our own MSDS sheet, which is available, and we create 4 parts per trillion. So most of us that are familiar with toxicological type or chemistry that's in the air, or anything that's over parts per million, we kind of shudder. If it's parts per billion, we don't worry about it too much. But 4 parts per trillion, how do you put that into perspective? That's what we produce. So when you think of 4 parts, and we're able to, with that 4 parts, control bacteria, virus, fungal spores, and VOCs, it's got to be pretty energetic. But when you look at, let's say, in the slide that you're going to have on your screen there, or in your books, an Olympic swimming pool, 4 parts per trillion would be one full drop in 5 Olympic-size swimming pools. So And what I'm trying to say with this is that there should not be any fear associated with this. When you're looking at hydroxyls, and the amount that's being put in is mimicking exactly what is being done in nature. So it's a very small amount in a very large area, but yet it's still able to do what it needs to do, and has been demonstrated to have been done over many, many years. So it's just to lay a few things to rest, because a lot of people have heard different things from time to time, and we just want to talk about the facts here, that it's 4 parts per trillion. That's our output. It's the same as in nature. And that picture puts it into perspective. Now, the next thing is validation. Who else has looked at this? So far, we've been pretty much talking about our work. And so when we engaged the Lovelace Respiratory Research Center, it was also validated again by Dr. D. Crosley, who's an atmospheric chemistry expert, and to even show that we created hydroxyls and the rate of production. So that was interesting. So our machines exactly mimic nature at that average of 2.6 million. So that was validated by Lovelace, that it was safe. So we had to do that at Comparative Biosciences, 18 pages, I believe, of credits to their how thorough they are. And in an FDA compliant laboratory, we just spoke about that. That was the rat study. So we know that it's safe for mammals. We actually know that we produce hydroxyls and at what rate. And also that we were able to reduce pollutants in the air, and that was verified by Columbia Analytical Services. And so the interesting thing is that we can deal with it in a building, or we can also deal with it just in stack emissions. Perhaps it's a very secure type of environment, and they just don't want us in the building. That's fine, because hydroxyls can travel with the airstream, the toxic airstream, and have those nasty components taken apart and neutralized by the time they get to the end of the stack. So that is an incredible advantage over technologies that require you to burn the air, in other words, incinerate the air, or force it underground into a biomass that it absorbs into it, and then you of course have to take the biomass away and burn it, or through carbon technology where you adsorb it onto a medium, or let's say you just blow it into the air but you throw chemicals along with it to try and, that is a wet chemical, to try and subdue those odors. So this is far more elegant. It does not require anything else but the ambient moisture and power to make it run. So got some wonderful advantage over what's currently there. And then the last test that we saw in the graphs were from ATS labs, and that we are able to subdue the hardest to subdue bacteria, virus, and fungal spores. And those tests are ongoing, as you can imagine, because as new microorganisms raise their head, we have to show that we can handle those as well. So that's a very powerful statement on one slide, that the Lovelace have their comments, the Comparative Biosciences, Columbia Analytical, and the biological end of things with ATS labs.